Okay, so this is going to be just a quick video on basically the overview of T3 and T4 synthesis within your cells. This is something that when I was a student, it was not part of the lecture. However, it's here for you guys, so we need to kind of learn it. So this is in lecture 13, I believe. Yeah, it's definitely in lecture 13. And it's basically just saying, what are these molecules used for? They're a very specialized amino acid, and they are derived from tyrosine, as you could probably tell from the structures just looking at them up here. So basically this slide up here, slide number 57, kind of puts the picture slide that follows it into words. However, I think that if I kind of explain this in a video, if I explain what's going on in the picture, it's going to kind of make it a little bit clearer, because this kind of helps you understand it a little better. And then obviously some of the applications of this are on the following slide, slide 59. But anyway, let's just get started with the picture. So the first step is that, because there's going to be a lot of transport of multiple things into and out of the cell, and the most important thing that we need to transport in first is going to be our iodide. Notice I'm saying iodide and not iodine. Iodide means that it is in the negative charge. That's the form of it right now. So you're going to make use of sodium once again. Now this is not the Na plus K plus transporter. This is a different sodium iodide transporter. So it's going to bring two of them in, and it's going to be symport. So you have to bring in your iodide so that you can bring it in from your blood and then into your thyroid cells, and then out into a different region where more chemistry can happen. This is a highly compartmentalized reaction. You can't have things all happening in one little space. It's kind of like the urea cycle, where some parts of it had to happen inside and outside of the mitochondria. So basically what's going to happen is that we are going to have our iodide and our sodium. We're going to take advantage of sodium to pump some iodide in. And meanwhile, your endoplasmic reticulum, let me get my pointer thing out. I forgot to do that at the beginning. So you're going to transport in the iodide and the sodium. And meanwhile, in your endoplasmic reticulum, which is very close to your nucleus, your endoplasmic reticulum is what does a lot of the sorting and packaging of different components in your cells. It can be a special protein that I can package and then send to its specific destination. All of you microbiome majors are probably going to know about this, but basically your endoplasmic reticulum sends things off in vesicles that are made of lipids. So it's sending off this molecule, which we'll talk about in a second, but it's sent off in a vesicle that has lipids on the outside. So as you can see, once it eventually gets to the membrane, the lipids of this vesicle just become part of the outer plasma membrane. But anyway, what's going on here is that your endoplasmic reticulum is sorting a really big protein called, it's abbreviated TG in this picture. And I crossed it out and wrote TG because there must have been a typo here and it was TB, and that's kind of not anywhere up in this explanation up here, so I assume it was a typo. But anyway, this proteins are derived from mRNAs that come from the nucleus, and then your ribosomes, which are on the rough endoplasmic reticulum, the ribosomes are going to synthesize that protein. So your smooth endoplasmic reticulum is going to package it and send it out. So step 2 is the TG secretion, and if we kind of zoom in on what TG is, it's called Thyroglobulin. Oh, whoops, I just accidentally drew instead of zoomed in. Let me fix that for you guys. So basically, if we zoom in on TG, we have to start looking at what it is. Uh-oh. Alright, the program is lagging, so I'm going to have to restart it. Let's take just a second. Alright, here we are. We're back. Alright, so I accidentally erased the TG, but this is TG right in here, so just keep that in mind. So we have to kind of zoom in and take a look at what thyroglobulin is. It's this protein, and I wish he kind of would have drawn this a little bit prettier. It's just a whole bunch of squiggly lines with what looks like a whole bunch of tyrosine sticking off of it. Now, granted, it is a globular protein, and you know, it's not going to have a pretty structure, but the part of the protein he wants you to just realize, because you have all this squiggly stuff on the inside. Let me get my pointer out again. You have all this squiggly stuff on the inside. That's just part of the protein, but you see all these tyrosines sticking off. And this OH group on the outside of the tyrosines is where all the chemistry is going to happen to eventually get iodine associated with it. So what's going to happen is that you're going to have to, this thyroglobulin protein is going to be sent out of the thyroid cell so the iodines can be added to it. So you have TG secretion, which is really right here, and we had to pump in the iodine, or the iodide, with sodium so that we can pump it out later. So the exocytosis of the thyroglobulin is not, it's transported, like the iodide is actively transported, but the thyroglobulin is just exocytosed from the cell. So now you have both your thyroglobulin on the outside of the cell and your iodide on the outside of the cell. So this is a region where chemistry can happen. So if we kind of scroll down a little bit, we're going to have to notice all of these, the thyroglobulin molecule, it has a whole bunch of aromatics, and there's an OH group on here, but that's totally fine. But if you think back to Orgo 2, how do you add things to aromatic rings? You have to do electrophilic aromatic substitution. That's EAS. So this is actually really bad to try to write it like that. I'm not even going to do it anymore. Sorry about that. My pointer is a little bit too thick to do that. So, but it's just EAS. So electrophilic aromatic substitution. We have to go through step 5, which is oxidation. So we're going to make this iodine lose electrons because it's in the I- form, so it has too many electrons. You need to change it back to neutral iodine so that you can do electrophilic aromatic substitution. If you have something that's negatively charged, it is a nucleophile. Nucleophilic aromatic substitution does exist, but your body does not use that in this mechanism. So that's what step 5, oxidation, is talking about. So now that you have iodine in a form that it can be electrophilically added to the aromatic ring, now we go through step 6, which is just iodination. And that's just basically saying, you remember how this happens in Orgo 2, there's, in the organic lab, you would have certain substrates and solvents you need to have with it, but we don't care about that anymore. So we're going to just add onto each of these residues two iodine residues. So these would be in the meta position, because this is like where I have my cursor right here. This is the base of the aromatic group, and then there's an OH group that is para to the base, and then one slot over here is ortho, and then 2 over is meta. So 2 over in this direction is also meta. So we're going to have 2 meta additions of an iodine, and basically once we have all of these iodines on here, we are now ready to go over through what's called conjugation. And conjugation is and he doesn't really say where these come from, but it seems as if the protein, the different tyrosine residues are plucked off from some other parts of the thyroglobulin. This is the whole entire protein. So he doesn't explain where it comes from. However, we have 2 choices. One, you just add random tyrosines from out of nowhere, or it's the tyrosines from other parts of this protein that are kind of cleaved off and then conjugated onto here. So I don't think you need to know it that in depth, but somehow we get these tyrosines to be added on. Now, what's interesting is that as we're looking at it right here, this is going to be T4, and this bottom is going to be T3. And there's a little bit of a discrepancy that I don't like about this picture, is that they can both form on here, but in the slide above in the words, it kind of tells you something different about how you get from T4 to T3, or maybe it's in the slide below. But anyway, so once you conjugate and you have, like, this is T4 and T3 attached to the rest of the giant globular protein, and we're only synthesizing this protein so we can eventually take off these key little parts of it, T4 and T3. So now that the protein has been modified on the outside of the cell, it can now re-enter through endocytosis, nothing too special going on there. And now the last couple steps, number 9, is proteolysis. So basically you have the T4 and the T3, which is what we're interested in, and then the rest of the globular protein is all over here. Proteolysis, you see the root proteo, so that means we're working with a protein, and lysis, which means we are breaking it apart. So as you can see, once we broke it apart, here's the rest of the globular protein that we don't want, and here are the useful things that we do want. Here we have T4 up here, which is Thyroxin is what it's called, and then we have T3, which is triiodothyronine. And obviously the last step is that you have some transporter. I don't think you need to know the name of this transporter, but it is pumped back out into the blood, because this is how you get these two useful molecules into your blood. They have to be synthesized in some special cell, in the protein factory of some special cell. In this case, it's a thyroid cell, a thyroid follicular cell. Just know it as a thyroid cell, though. So once you take care of making that, they can go out into your blood, and then this molecule can do useful things. And it kind of explains what it does, T3 and T4 in your cell. It explains it in the slides above and below this one. But so what I do want to say, the last thing I'm going to say, is that there is a way for your body to take T4, which is right up here, thyroxine, because we see that it has 1, 2, 3, 4 iodines on it, and then there is a way to get it to 3 iodines, and that, I believe, is on the next slide. Let's see, no, it is on the slide above that, I believe. Let's see. It was written somewhere in here, but basically there is just like we cleave phosphates off the side of ATP, there is a way we can cleave off just one of these iodines. No, I think it is on the bottom slide. Let me go back to that. Oh yeah, it's just right here. Okay, so I apologize about that. T3 is formed from T4 by a peripheral deiodinase. So if you take a look at the name of this enzyme, deiodinase, we see ACE right here, so we know that it's going to be some type of enzyme, and usually enzymes with ACE, they're going to break something down, but just look at this. We are deiodinating something, so the name kind of tells you what it does. Once you make T4, you then can cleave off just one of them to make T3, and it says that T3 is much more active in our cells. In all of our cells, our target is for T3. However, T4 and T3, they have slightly different functions in the body, and these little bullet points down here, they just tell you what it's useful for, what T3 is useful for. I know up in here it says that T4 is going to be, it's actively transported. We talked about that, but it has a lot to do with your central nervous system, which is what CNS stands for. So basically, that's all we want to go over in this little video. But yeah, this picture right here, and once again, there should be a TG right here. That was had a little typo in his notes, I suppose. But yeah, that's basically what you need to know about this. And I would, now that you understand this picture, he's probably not going to ask you about too much of this process, these steps of how your cell does it. since this is a clinical case, which that's why you see this little symbol over here, it's on all these slides, he's probably going to favor asking you questions about stuff on slide 59. Obviously, you need to know kind of everything in this course, but this is where I think the test questions are most likely to be formed from. So, yep, that's it for this video, and let me know if you have any questions.